So this is going to plug my doctoral work a little bit, which is the treatment of mastitis in dairy cattle. So mastitis is an inflammation of the mammary gland, and it's a production issue in the dairy industry. So the infection is caused by Staphylococcus aureus, or considered the most problematic in the sense that they don't treat, well, they don't cure well with antibiotics, and also they shed Staph aureus into the milk, which is never good. So we did some preliminary work. We ended up conducting an animal trial and had a placebo-controlled trial in 18 naturally occurring mastitis infections. So this is not challenge. These are actual, real Staph aureus infections that were acquired in the wild, and we had a group of 20 in a failing placebo control. Now we did have 3 cures in the treatment group, but it's not statistically significant. And we'll talk about that in a minute, but it does show the difference between doing work in a mouse model and doing work in the real world. That's my one. So there's been some work to look at phage therapy in aquaculture. So where the fish are all jammed together, and there are several diseases which will run through the fish tanks. So particularly this one paper here, or sorry, series of papers where they looked at Lactococcus garbii and Pseudomonas lectococida, if I pronounced correctly, which are both major fish pathogens. So they saw good efficacy when they administered the phage early. This is again challenge trial. This is not natural infections. But again, this is kind of a recurring theme we see in phage therapy, that if you treat them right away, you tend to see very good results. So if they treated them at 0 or 1 hour after a challenge, then you have high survival. And if you wait 24 hours, you get less survival. And of course, if you don't treat them, you get really bad survival. So, yeah, so as I said, this is a common theme in phage therapy, and it clashes it with the theory. In some cases, it's almost like the dogma of phage therapy, which is that the phages can replicate on their hosts, and so you don't want to treat them very high with a very high dose of phage, because you don't want to wipe out the pathogen population too quickly. You want the phage replicate in that population, develop into even higher numbers. But that theory does not hold up very well in practice, and that delayed treatment, and this is true in several other studies as well, that if you delay treatment for phage therapy, you tend to see poor efficacy. So one model for this is that there are populations of pathogens which are protected in the host, and so they never see a phage. And then once the phage are eliminated from the system, as they will be eventually, then they can reemerge and they can recolonize the host. So this is some work we did looking, this is based on the results of the mastitis trial that I showed you. So we fluorescently labeled the milk whey protein. Of course, this is a treatment of mastitis, the mammary, so it's full of milk. So we labeled these whey proteins with a fluorescent, so amine reactive or fluorescein dye, and then incubated that in the cells, Staph aureus cells, in that. As you can see, they all pick up a very nice coating of that protein. And so this provides, I think, a pretty good mechanism of how you get phage, how you get bacterial escape from phage attack. That at least in this system, which is the bovine mammary, the bacteria they pick up these coats of host protein absorbed to the surface, and this includes phage attachment. And you can see here, if they're grown in this protein, they also form these very large aggregates. And this is actually a smaller one of the aggregates that you can find. So it's almost like a flocculent biofilm that they form inside the gland. And this is some SEM of similar exposure. So this is the unexposed, BSA-exposed Staph aureus cells on a field emission microscope, electron microscope. And as you expose them to the protein, they pick up this layer of debris and also a notable roughening of the surface. And to further complicate things, of course, this is like a real infection. They're in a real gland, so the bacteria are up here somewhere, and the tissue structure is very similar to a lung. You have these alveolar sacs. And you're administering the phage down here. So your phage has to travel all the way up through what is undoubtedly meters of ductwork and to reach the bacteria which are covered in protein and stuck to the side of some of these alveolar sacs. And so this is the difference between a challenge infection, which you can treat immediately where the bacteria haven't really had much chance to become established, and an actual infection where the bacteria have been there for perhaps weeks or months, and they are well established in the tissue, and then you have very poor penetration. So a common concern with phage therapy is that you'll have resistance to the phage, and this was brought up, very early on in the 60s. There's a few papers and also in the early 70s. And this is one of the major concerns. The reason why phage therapy will never work because you'll put that in, you'll put your phage in there, and the bacteria will become resistant, and then everything's ruined, and you'll have to just give up, and the person is hopeless. But that's not exactly true. Now there are some studies where they have done their phage therapy experiments, and they end up with some efficacy or actually not very good efficacy, and then they isolate the bacteria out of those animals, and they have resistant cells. So you reculture them out of the challenged animal, and you grow them up, and you plate your phage on them, and you find that they're resistant to that phage. And that does happen. But for instance, in the work with mastitis with Staphylococcus aureus, we cultured on every animal that failed to cure, cultured the bacteria out of them, and they're all still sensitive to the phage. So there are other mechanisms that you can have treatment failure that are not resistant. And additionally, if you have resistance, there are some systems where you'll get the bacteria incur a large fitness cost if they become resistant. And this is exemplified in the work that was the Smith and Huggins work in Chap diarrhea, where they used the E. coli K strain as E. coli K1, which causes the diarrhea in the calves, but its major virulence factor is the capsule, which is also the phage receptor. So if the bacteria mutates to become resistant to the phage by losing its capsule, it also becomes avirulent. So in that sense, the phage actually, the bacteria can't really win. So it's either lysed by the phage, or if it becomes resistant, then it's eliminated by the host, because it's no longer able to protect itself from the host immune response. So this has been some work also done in plant agriculture with phages. So there's a group in Florida, Jeff Jones, his group, who have done some work with treating tomatoes, also peppers, Xanthomonas bacteria. And so they were able to get significant control versus the standard treatment for their copper and mancozeb. And in plant agriculture, this is always useful, actually, and particularly now, because the antibiotics which are typically used for treatment in plant pathogens are often they're being delisted for use in agriculture. So I used to do work in apples. The standard treatment, which is recommended for say treatment of fire blight, was they would sell these 1 kilogram bags of streptomycin, which you mix into a tank of water, and you go spray that out over your orchard. Now you're not allowed to do that anymore, and so there are requirements for alternate treatment methods for plant pathogenic bacteria. So they also use a strategy of host range, generating host range mutant phage. Right here. So this is another strategy for getting around bacterial resistance. This is also something which we've been able to do with the Burkholderia phage in our lab. So you have your original bacterial strain here, which is sensitive to the phage. Of course, it'll mutate to become resistant at some point, which is this one down here. And so it'll then multiply and take over, so you have this resistant bacterial strain. However, you can simply use the capacity of the phage to mutate to locate isolated phage, which is able to overcome that resistance. And you'll have a phage that's able to lyse this resistant strain, and then you can propagate those phage on that strain, and then you'll have a new phage which is able to lyse the resistant strain. Does that make sense? Right. So you can actually continue to do this for a few rounds, although another interesting point for looking at bacterial resistance is that people tend to think of it as the arms race between phage and bacteria, and you mutate, counter-mutate. But if you do this in relatively closed systems, it's not an infinite cycle. So there has been some work done, say, in chemostat culture, where if you challenge phage and bacteria together, every mutation that every mutation that the bacteria acquires to become resistant to the phage also incurs some kind of fitness cost on the bacteria. And so you can only have this kind of phage bacterial arms race go for 2 generations before the bacteria, the resistant bacteria, become unfit, and they're actually less fit than their phage-sensitive parents. And so they're actually better off being sensitive to the phage than to live on in this incredibly unfit state. And so it only tends to go for a few generations. In the chemostat culture, I think it went for pretty much like 3 cycles before the bacteria were so sick that they really didn't compete in the culture anymore.